Hey everyone, today I'm going to talk to you about mitosis and meiosis.
Yeah!
Shut the heck up, kids. I have to teach you something, and you're going to learn it.
You don't have to be so mean about it.
First things first, the cell cycle. What is it? Here's a pic.
Check this out. The cell cycle is 90% interphase, 10% M phase. In interphase, we have the G1 phase, the first growth stage, the S phase, where chromosomes prepare to duplicate, and the G2 phase, the final growth stage. After that, we enter the M phase, made up of mitosis and cytokinesis. Pretty cool, huh? Faux shizzle, my nizzle.
I don't need your approval, Stoop Lion. If I needed Stoop Lion's approval, I would have asked for Stoop Lion's approval. You dig? Now let's get on to something more important here, like mitosis. This next one's a doozy. Bear with me, folks. An assistant, bring me my caffeine.
Holy cannoli. This is gonna be good. In interphase, the nuclear envelope encloses the nucleus. Two centrosomes are formed by the duplication of a single centrosome. Each centrosome contains two centrioles. Chromosomes duplicate during S phase, but they can't be seen individually.
Slow down!
Silence! Remain obedient to the lesson. Now on to prophase.
In prophase, chromatin fibers tightly coil and condense into chromosomes. The nucleoli disappears, and chromosomes appear as two identical sister chromatids join at centromeres. Mitotic spindles begin to form. Centrosomes move away from each other. Microtubules extend from each centrosome. Chromosomes are more condensed. Each chromatid has kinetochores and some microtubules attached to them. Non-kinetochore microtubules interact with these chromatids from opposite poles of the cell. What you talking about, Glover?
Let Gary Coleman in here. Metaphase.
During metaphase, centrosomes are at opposite poles of the cell. Chromosomes meet at a metaphase plate that is equally distant between the poles. Chromosomes, the kinetochores of sister chromatids, are attached to kinetochore microtubules coming from opposite poles. Hey, what comes next? Anaphase, the shortest stage. Cohesion proteins are cleaved, allowing sister chromatids to part from each other, becoming a chromosome. Daughter chromosomes move to opposite ends of the cell. The cell elongates, leaving an equal number of chromosomes at either end of the cell.
In a minute, you're going to have to know what cytokinesis is. So, what is that? It's a separation of the cytoplasm, if you will. Animal cells experience a pinching off that looks something like this.
Crocky, mate. There's an animal on the loose.
Plant cells undergoing cytokinesis experience the growth of a new cell wall. Like this.
Let's get down to business. Talk about telophase. During telophase, two daughter nuclei form the cell, along with nuclear envelopes and nucleoli. Chromosomes decondense, and the remaining microtubules are depolymerized.
Doesn't that blow your mind? One cell just turns into two. You better bring your umbrellas, folks, because it's raining knowledge. Now that we're done with mitosis, let's talk about meiosis, which produces sex cells.
Just groovy, baby, yeah.
Quiet, Mr. Powers. Quiet.
Meiosis 1 is a separation of homologous chromosomes. In prophase 1, duplicated homologous chromosomes pair and exchange segments. In metaphase 1, chromosomes lined up by homologous pairs. In anaphase 1, each pair of homologous chromosomes separate. In telophase 1 and cytokinesis, two haploid cells form. Each chromosome still consists of two sister chromatids.
This is getting hard!
Shut up, kid. It's not my fault you can't read. Who the heck are you?
Meiosis 2 is a separation of sister chromatids. In prophase 2, a spindle apparatus forms. Chromosomes each still composed of two chromatids associated at the centromere. They move towards the metaphase 2 plate. In metaphase 2, chromosomes are positioned at the metaphase plate and are genetically alike because of the crossover in meiosis 1. In anaphase 2, a breakdown of proteins allow chromatids to separate. Chromatids move to opposite poles as individual chromosomes. In telophase and cytokinesis 2, nuclei form. Chromosomes decondense and cytokinesis occurs.
So what does this all mean? In mitosis, two identical daughter cells are formed that are both identical to the mother cell. If the parent cell is a haploid cell, both the daughter cells will be haploid cells. If the parent cell is a diploid cell, both the daughter cells will be diploid. Simple. Now in meiosis, one parent cell produces four daughter cells, each with a haploid of unduplicated chromosome. Four daughter cells are generally distinct from another and from the parent cell. That's it. You got it now?